Hello and welcome to the ASCO Daily News Podcast. I'm your host, Dr. Monty Pal. I'm a medical oncologist, professor, and vice chair at the City of Hope Cancer Center in Los Angeles. And today I'm super excited. We're actually going to be covering some of the developments at the most recent ASCO Breakthrough Meeting. We have a range of topics from targeted therapies to immunotherapy combinations in real-world settings to circulating tumor DNA that we're going to be discussing. And here with me today is just the perfect guest, Dr. Roselle De Guzman. She's a visionary leader in global oncology, and she's actually chair-elect of the 2026 ASCO Breakthrough Program Committee. So who better than to discuss these exciting abstracts than her? Dr. Guzman is also an associate professor and medical oncologist at Manila Central University FTT Medical Foundation Hospital in the Philippines. I also want to note that she was past chair of the ASCO Asia Pacific Regional Council, and she was also a member of the ASCO Scientific Program Committee previously. So a very deep bench of ASCO experience. Our full disclosures are available in the transcript of this episode. Dr. De Guzman, welcome and congratulations on such a fantastic ASCO breakthrough in Singapore.
Yes, thank you very much, Dr. Pal, and thank you for having me here. It was a real privilege to shape this year's program, and even better to see it come together in the room with so much energy from the region.
I have to say, I was following very, very closely on Twitter. I had an excellent opportunity to view many of the abstracts, but I didn't really do a deep dive. Now, there were over 300 abstracts presented at the meeting, pretty huge meeting, and it was a great opportunity to really inspire collaborations across oncology, industry, and tech. I wanted to focus on a couple of the abstracts that really caught my attention. And the first of these is abstract number 14, in case you're following along and your home audience there. And this is an abstract dealing with real world performance of a multimodal cell free DNA multi cancer early detection test in Asian populations. I know that's a mouthful, but this was actually presented by the group from Medical Genetics Institute in Ho Chi Minh City. Dr. De Guzman, do you mind just giving us a little primer on what this abstract was all about?
Yes, sure, Dr. Pal. So, this abstract studies the real world evidence looking at the multi cancer early detection test in an Asian population. So, the assay, SPAT-MAS, which stands for Screening for the Presence of Tumor by Methylation and Size, is a liquid biopsy assay integrating methylomic, fragmentomic, and genomic signatures of cell free DNA to detect signals across 10 cancer types. So it had already been validated in a smaller prospective trial. So what's new here is the scale and the setting. Over already been tested 84,000 people with 22,597 asymptomatic individuals followed for a full year across 6 countries in the region. And so participants with positive signals underwent standardized consultation and workup with imaging or histopathology according to a predefined protocol. So we're not just looking at a lab number, we're also looking at confirmed clinical outcomes. And one third of those diagnosed with cancers were those types of cancers that lack standard of care screening options. So the SPAT mass has a sensitivity of 79%, specificity of 99.9%, and a tissue of origin accuracy of 80%. So this study provides the first large-scale real-world evidence from Asia supporting the robustness and clinical utility of SPAT mass as a complementary screening strategy. So, a single blood draw can flag multiple malignancies, including ones those we would otherwise catch late, and could potentially change the approach to cancer control, provided the cost as well as the diagnostic capacity are addressed alongside it. So, the timing of this data is worth noting too. Just weeks before the breakthrough meeting in Singapore, SPATMAS 10 received US FDA breakthrough device designation. So, this real world Asian cohort isn't just a regional validation story. It adds in advancing accessible, evidence based technologies that support earlier detection of cancers, especially for those where screening options remain limited. So, that will ultimately help to improve patient outcomes worldwide. So, just to say also that SPATMAS is designed to complement and not replace the existing guideline recommended standard of care cancer screening tests, while At the same time, it expands early detection opportunities for cancers that lack established screening pathways.
I think that's a brilliant summary of a very, very large and complex data set. This is one where I really want to sort of dig into the weeds and better understand. But as you think about the broader population there in the Philippines or perhaps in Vietnam, where the study was reported out of, is this the sort of test that's going to be just available to any patient who walks in the door? Or what would be the profile of who you might offer this to?
Well, of course, when we're talking about a blood based test, this is very attractive for patients and individuals, especially those who have high risk factors. But we have to consider also the limitations in terms of the infrastructure and healthcare systems. The region, the Asia Pacific, has a diversity in the healthcare infrastructure and systems. What could be happening in Hong Kong, in Singapore, are very different in what's happening in, as you mentioned, in Vietnam and in the Philippines. So definitely not all countries in Asia Pacific do even have the standard national cancer screening programs. But the development of these types of real world multi cancer early detection tests may move the landscape forward in such a way that we may have an in-depth communication and collaboration with our stakeholders, the government, the ministries of health, in order to convince them and let them know that we are moving not from the traditional cancer screening programs into a more advanced multi-cancer early detection test. So in the current setting, in the current landscape, we do have access to, let's say, mammography for breast cancer, CT scan for lung cancer, colonoscopy for colorectal cancers. But there are limitations because there are types of cancers like head and neck cancers, esophageal, pancreatic, ovarian that are increasing in incidence. And these types of cancers lack the currently standard screening methods. And tests like this or SPATMAS multi-cancer detection test may fill that gap of expanding early detection opportunities for these types of cancers as well.
Yeah, absolutely. So, we're going to kind of stay on a similar theme here to Spot Mouse, and we're going to shift gears, but maybe focus a little bit more on colon cancer. This next abstract that I wanted to cover, this is number 94, comes from Dr. Sun and colleagues from the First Affiliated Hospital of Jinan University in Guangzhou, China. And this actually has to do with cfDNA methylation using clinical parameters and this novel test alongside AI in really defining precancerous and cancerous lesions in colorectal cancer. This is a complicated abstract. Walk me through it.
Yeah. So the investigators enrolled about 1,370 participants, and these are patients with colorectal cancer, those individuals with precancerous lesions, high risk individuals, and healthy controls. And they combined cell free DNA methylation markers with biochemistry factors like tumor markers, white cell counts, and also clinical factors including age, smoking, and metabolic parameters. So they tested 15 different AI algorithms and landed on AdaBoost as the best performer. And so by cell-free DNA, both by electrochemical absorption and colloidal-based absorption, these two techniques they used at the same time, they found that colorectal cancer sensitivity is at 98% and specificity of 99.8%, and precancerous lesion sensitivity of 89% and specificity of 63%. So this study was developed using enriched case control Asian cohorts. So this needs prospective validation across different populations and ethnic groups. It needs validation in average risk population and compared against colonoscopy ideally. But also demonstrate feasibility and should also demonstrate cost effectiveness prior to implementation in different health systems. So, early detection in precancerous lesions and early colorectal cancers, we know this reduces colorectal cancer mortality. blood-based cell-free DNA methylation screening platforms have progressed beyond the single gene to be multimodal or multidimensional with improvement in precursor lesion detection. So we need prospective validation of this in average screening population.
I like that approach. And I completely agree with you. I mean, understanding how these function in different populations and so forth is really, really critical. We're going to shift gears again, and this is a bigger shift. And we're actually going to talk about some prospective data that was presented at the ASCO Breakthrough Meeting. And this comes from the INAVO 120 phase 3 clinical trial. So this was actually looking at the context of breast cancer and specifically patients with PIK3CA mutated, hormone receptor positive, HER2 negative, endocrine resistant advanced breast cancer. And these patients were treated either with inavolisib or with placebo or palbociclib with fulvestrant. And maybe you can give us a little rundown of the specific objectives and focusing on an Asian population here.
Yeah, sure. So this abstract asks the question we always need to ask. Does the benefit hold up in an Asian subgroup where body composition, metabolic risk, and mutation prevalence can differ from the global trial population? So the INAVO 120 included 120 Asian patients, and it showed that the median progression-free survival was 14.8 months with the inavolisib and palbociclib and fulvestrant arm versus 6.8 months with the placebo, palbociclib and fulvestrant arm, and with a hazard ratio of 0.38. Median OS was 32.7 months versus 27 months with a hazard ratio of 0.74. The response rate more than doubled at 60% versus 27%. And for the adverse events that were more common in the Asian population, cytopenias occurred in at least 20% of patients in either arm. The frequency was relatively similar for all grades in both the inavolisib arm and the placebo. But the frequency of all grade hyperglycemia, diarrhea, stomatitis, nausea, decreased appetite, and fatigue were numerically higher in inavolisib arms. Most of these adverse events were grade 1. So, additionally, on safety, hyperglycemia was the most common reason for dose interruption and discontinuation rates. And they were numerically lower than in the overall trial population. So, consistent with the overall population, inavolisib and palbociclib and fulvestrant showed a clinically meaningful efficacy benefit in Asian patients with no new safety signals and numerically lower discontinuation and dose reduction rates versus the intent to treat population. So this is important for us because for Asian patients that we see on a day-to-day basis, this gives us real reassurance that the regimen's benefit risk profile translates, and it reinforces why we should be testing for PIK3CA mutations routinely in this setting. You know, Dr. Pal, a global trial can tell us that a drug works, but a well conducted subgroup analysis like this one tells us that it works also for the patient sitting in front of us, whether we're holding clinic in Singapore, in Hong Kong, in Bangkok, in Manila. And that distinction matters when you're the one having the conversation with the patient.
I think that's a wonderful, wonderful takeaway from this is that virtually all the phase 3 studies that we're doing now leading to FDA approvals and then implementation worldwide, it's so critical that we try to parse out the data as best we can by region. I'm guessing you and I are on the same page, right, Dr. De Guzman? Is this something that you'd like to see just sort of across the board for the majority of phase 3 studies that we're running across oncology?
Yes, completely agree.
Yeah, absolutely. Moving from prospective studies, I mean, I think the other way that we can really sort of garner some evidence around how drugs work in our populations is through real world data. And the next abstract, 135, comes from Dr. Biswas. And this is a group effort across actually several continents here. So we have investigators from the All India Medical Institute in Jodhpur, India, from New Delhi as well. We have investigators from Ukraine and even Sudan coming together to report data that looks across several treatment regimens for metastatic esophageal cancer, specifically looking at frontline therapy with chemo pembro, chemo nivo, and then nivo ipi. Would you mind walking us through this abstract? This is abstract number 135, by the way, for those listening at home.
The combination of pembrolizumab and standard chemotherapy was from the Keynote 590 trial, just to refresh our listeners. So that was the first to show a significant improvement in survival, regardless of PD-L1 tumor status in the first-line treatment of esophageal cancer. And then the Checkmate 648 trial is about first line nivolumab plus chemotherapy and nivolumab plus ipilimumab, which both demonstrated significant overall survival benefit versus chemo in previously untreated patients with advanced esophageal squamous cell carcinoma. So Pembro with chemo from Keynote 590 and nivolumab plus chemo and nivolumab plus ipilimumab from Checkmate 648 are all approved as first line treatment. So the investigators used a large federated real world database from global collaborative network of 171 organizations, and they did propensity match comparisons across all three regimens. So the chemotherapy containing regimens performed comparably to each other, but the nivolumab plus ipilimumab showed a meaningful overall survival advantage over nivolumab plus chemotherapy, 443 days versus 303 days, and a favorable trend against pembrolizumab plus chemotherapy as well. So, just as importantly, neutropenia rates were markedly lower with the chemotherapy free combination, and there were no differences in colitis, hypothyroidism, nausea, and vomiting. So, this real world propensity match data does support what many of us already suspect clinically, that a chemo free immunotherapy doublet is a viable first line option, particularly for patients who are older, who are frailer, or at higher risk for hematologic toxicity, which describes a meaningful proportion of the esophageal cancer population that we see in our clinics.
And maybe tell us how this applies in your microcosm in the Philippines. Would you maybe have more of a lean towards Nivo-IPI, for instance, after looking at this data, or from a practical implementation perspective? Is this data translatable?
Yes, definitely. The types of patients that we see, not all of them may be able to tolerate the effects from chemotherapy or the chemotoxicities. However, one thing that we have to consider is the access, the cost, because the healthcare system is not always reimbursable, and a majority of our patients pay out of their pockets. So, one thing that might be a barrier to apply this in clinical actual practice is the cost issue and the access.
Indeed. In terms of practical elements of treatment, this sort of brings us to the last abstract that I wanted to go through with you, which is abstract TPS 293. So, this is one of our trials in progress abstracts, but it kind of covers a unique topic and something we haven't chatted yet about on this podcast, which is adabrelumab and oral etoposide, specifically in older patients with small cell lung cancer. I can imagine this is a very vexing issue. Small cell lung cancer is obviously an incredibly aggressive disease with, I'll say today, limited treatment options. Tell us about why this trials in progress abstract stood out to you.
Yeah, this is a trial that speaks directly to a population we don't always design trials around. So we all know that older patients and those with poor performance status or significant comorbidities are usually ineligible for standard small cell lung cancer trials, even though they make up a significant number of real world cases. So we learned from Capstone 1 trial. So this is the regimen debrelumab, which is an anti-PD-L1 monoclonal antibody plus platinum etoposide, that combination significantly improved median overall survival compared to the standard chemotherapy as a first-line treatment for extensive stage small cell lung cancer. So the addition of this immunotherapy alone showed strong activity in the first line setting, but a lot of our patients simply cannot tolerate a platinum chemotherapy backbone. So this phase 2 study substitutes oral etoposide for the platinum doublet, pairing it with adebrelumab in patients who are 65 and older, who have equal performance status of 2 or 3, and an expected life expectancy of at least 3 months and adequate organ function. So, additionally, those patients maybe age less than 65 with an echo of 0 to 1 may be considered as well if they have significant comorbidities. So this trial is enrolling 47 patients, treatment naive, across multiple centers in China, with progression-free survival as the primary endpoint. So the oral chemo backbone may be more feasible in some settings, and this may allow us to bring immunotherapy combinations to patients who would otherwise be offered best supportive care alone.
It's such an interesting concept. It actually gets me thinking about my very first mentor in oncology, Arti Huria, who was, one of the pioneers in geriatric oncology, I would say. She always had such a focus on better understanding what we could do for our older patients to specifically address their needs. And certainly this trial very much speaks to that. I mean, it's a really exciting design. I have to say, Dr. De Guzman, thank you so much for all your incredible work this year. It seems like an incredibly robust ASCO Breakthroughs meeting. Any closing thoughts you wanted to offer before we wrap up?
Thank you very much also, Dr. Pal, for having me here. I believe that's the real mission of ASCO Breakthrough, and that's why an event like this one matters so much to us practicing in Asia, because this meeting has a regional focus, and I'm grateful to the presenters, to the researchers, to the investigators for doing what they do and also sharing their work with us. And I'm already looking forward to seeing what comes out of next year's meeting.
Indeed, indeed. Well, thank you so much for being here. And thank you to our listeners, too. You'll find links to all the abstracts we discussed today in the transcript. And finally, if you value the insights that you heard today on the ASCO Daily News podcast, please take a moment to rate, review, and subscribe wherever you get your podcasts.
The purpose of this podcast is to educate and to inform. This is not a substitute for professional medical care and is not intended for use in the diagnosis or treatment of individual conditions. Guests on this podcast express their own opinions, experience, and conclusions. Guest statements on the podcast do not express the opinions of ASCO. The mention of any product, service, organization, activity, or therapy should not be construed as an ASCO endorsement.